Welcome everyone to our Science Thursday with Brookhaven Lab. My name is Alita Perez. I am from the Office of Educational Program, and I'm joined by my colleague Diana Murphy, who will be managing the Q&A section for our discussion today. Before we start, just a few reminders. You can submit your question using the Q&A chat session, and that is on the right side of your screen. And we will try to get as many questions as we can today. If you have any issues or difficulties with the video streaming, please feel free to also send the IT know by making a comment on the chat section too. So today I am joined by Jantana Keereetaweep. She's a biochemist at Brookhaven Lab in the biology department. Welcome, Jantana. How are you?
Good. How are you?
Well, thank you. Thank you for joining us today. I am so
glad
Thank
you for having me here.
I mean, I think biologists are the best, and so thank you.
So hopefully at the end of today, the students will think that biology is the best too, right?
Yeah, they will, and they can also get a flavor of the various things that is done in biologists do. And then know that the interdisciplinary aspect of that, right? You can be a chemist and still do some work in the biology fields, which is great. So yeah. I learned recently that you won the Paul Stump Award, and so for our guests, can you tell a little bit about what that award is all about?
Yeah, Paul Stump Award. First of all, I feel really honored by it, and I want to thank everybody that's being the biggest part of this award. All my lab mates, my supervisor, Dr. John Shanklin, the biology department, and actually the whole Brookhaven lab. All my collaborators that did such a great job in all the experiments, everything that was leading to this point. So the Paul Stump Award is the International Plant Lipid Researcher. They give this award every other year to the early career scientists. So yeah, so yes. So this year, yes. Yeah, so every other year. So this year, actually, the meeting was going to be in France, but because of the pandemic, so we're going to just give the lecture online in July. Yeah.
Awesome. Is that lecture will be able to? Is a lecture that everybody can see and join?
Yes. Yeah.
Okay.
Yeah.
Awesome. Yeah. So I'll catch you after we're later on. I'll reach out. Maybe we can share that link with our students as well.
Absolutely.
So we're gonna play a video that is gonna start our conversation, and this is a video that BNL put together when we made the announcement that John has been awarded with the Stump Award. So Gabe is gonna put it up, and it's about a minute or so.
Plants have amazingly diverse metabolism, and we can study that and learn about it and modify it to enable plants to make essentially any of the products that we can use as a society.
We want to use all the resources already available in the plant cells to manipulate plants to produce sustainable energy and other desirable products. We're trying to understand the mechanisms in which protein responsible for oil production in plants interact at the molecular level, and then use that knowledge to manipulate plants to make more oil. We have growth chambers, we have greenhouses, we have incubators to grow bacteria. We also use the National Synchrotron light source to use the X-ray beam to study the molecular level of how proteins interact. Plants are the future. We want to manipulate plants to make more renewable energy and other desirable products.
That is great. So Just taking from that video, Jantana, can you tell us a little bit about more about the research that you do here at the laboratory?
So basically, in the nutshell, we're trying to make more oil in plants. The goal is that, make more oil in seeds or make more oil in leaves, vegetative tissues of plants, right? Mm-hmm. So when you think about, plant oil, everybody will think about, yes, we'll collect the seeds, extract lipids from seeds. But then when you look around, look outside your window, what do you see most? It's actually leaves, right? Mm-hmm. So leaves is the major, it actually make up the most mass of the plants. So we're also thinking that, how do we get leaves to make fat, to make oil as well? Mm-hmm. Okay, so I would love to share my screen here a little bit, just a little bit to show you guys, what we're using. Share screen. Entire screen. Start sharing. Okay. So can you see the screen now? I hope. Right. Okay. So that's the model plants that we're using. So this one is Arabidopsis. They're really, really small. If you can see the scale on here, the plant is actually really, really tiny. The seeds of these plants is so small that, if you don't look carefully, you can't see them. So the advantage of using this plant is that it grows really fast. Okay, the life cycle is really short. So you grow the plants, and then you can collect the seeds in a few weeks. And the genome is completely sequenced, so we know exactly what genes we're dealing with and where they are located. So that's another model plants that we're using is brassica. We probably familiar with the rapeseed oil, so that's what we're using as well. This picture, just want to show you guys a little bit here, the kind of summary of what we're doing, what we're looking at. So this is a complex that's responsible for the first step of fatty acid synthesis. So the lipid biosynthesis in plants or in mammalian system, these are really, really complex. It's intricate. The regulation of the lipid biosynthesis is tightly regulated. So you have so many pathways involved in lipid biosynthesis. So this is one that we were looking at. So this complex, you can see that is a large protein complex that has 4 different subunits. And what we were looking at is that we use the system that you actually feed excess fatty acid into our model plant. In this case is the tissue culture of Brassica napus, which is this one I show you in the diagram here. So we want to see that, hey, in case that plants already produce a lot of fatty acid, plant, what is the process? You just want to stop making anymore, right? Because you sense that, hey, I'm already fat. I'm going to stop making fat, right? So it's just like, hey, you know what? Plants already fat. I'm going to stop making more oil. But I'm evil. I'm going to trick them that, hey, I want you to get fatter. I want you to make more oil. How are you going to do that? So this is one of the things that we were looking at. My colleagues and I has been working on this. So we saw that this giant protein complex, it actually has the inactive analog that actually can be incorporated into this giant complex and make the complex inactive. So in case, in the situation that you have really high fatty acid, this inactive analog will go into the complex and make the complex inactive. What does it mean? It means that, okay, it's going to halt lipid biosynthesis. So what we were looking at is that, hey, what if we just get rid of this inactive subunit? What's going to happen? So we, my colleague and I, looking into this. So we knock out this this protein. Okay, now the plant won't produce this protein anymore, so it won't be incorporated into the complex. Even when you have high fatty acid, what does it mean? It means that plant can't tell that, hey, you know what, I'm going to stop making more oil. So the end result is that we can produce more oil in seeds.
So you're tricking the plant to sort of to think, so you're changing the way that the molecular biology of the plant, so the plant signaling pathway, so the plant just basically can continue making oils without having been able to say, I'm sorry, and there's no stop signal. So they're getting bigger. I would use the word bigger or bigger and bigger. They're just accumulating more and more and more oil, right?
Right, exactly. So it's just like, you're driving a car, okay, and you just remove the brakes, you don't put brakes on anymore. Just go full speed, make more oil.
So how is the plant figures it out? How the plant knows how the plant figures it out that is there any way that the plant senses when that fatty acid needs to be made or not, or to stop that production or not?
Can you repeat that again, Alita? Sorry about that.
So the plant, so you have a pathway where the plant just simply stop making it. It makes the oil without, continuously. When would you want to stop that? When it gets to a point, how do the plant in real life senses that signaling, that pathway?
That's a great question. So another example that I want to show you here is sugar signaling that's actually regulate lipid biosynthesis as well. So we know that, hey, when you have a lot of sugar, what does sugar do? It is the carbon skeleton, right? So it's actually feeding the carbon skeleton into the system. What are you going to do with that? You can make oil out of that, right? You can make fatty acid out of that. So I'm going to share this real quick with you
it's
guys.
Like humans, right? When we eat a lot of sugar in our body, we store that sugar as fat molecules in our skin.
Right. Share screen. Okay. Now I'm getting really savvy with sharing the screen now. So you guys can see the screen now, right? I hope. Yes. So this is technology that we've been using. It's actually really, really cool. The instrument here, very small actually. If you can see in the little picture here, can you see my cursor though? Yeah. So you see that it's a tiny capillary right here. So you dip the capillary into your reaction. You can use just, I don't know, 4 microliters of your reaction.
And what kind of reaction is that, Jen? That is that
basically for this particular study, we study the direct interaction, protein-protein interaction, and also protein and small molecule interaction. So like I mentioned earlier, that we also study the sugar in regulation of lipid biosynthesis. So this is what this is the instrument that we use. It's called microscale thermophoresis. The reason that we use this instrument is that it's very simple, it's very easy. So you have, let's say that you have 2 purified protein. You label one protein, label with fluorescent dye, and you have another protein which is not labeled. So basically, you mix them together, right? So you have one control, you have one of your experiments. One control is, whatever buffer that your protein's in. And another one, you have your 2 proteins together, or you have one protein and one small molecule together. So the movement of the protein when you have the laser in this instrument on, the movement of the protein, one protein alone, or the protein complex, or the protein with small molecule, they move differently. They move differently when they are the laser hit, right? When the laser is off, they will go back to their normal state. differently as well. So here you can see that the complex, right? So you have size different, hydration shell different, and charges different, right? So these are the factors that we monitor, okay, that will make a difference in the movement of these protein complexes. So we have a series of the reaction, and we just follow the movement of the complex or the protein, the one protein alone, basically. So what we found from this study is that so when plants has, high sugar, a lot of sugar, okay, so it will send the signal. Okay, so it will send the signal that, hey, you know what, you have a lot of sugar. What are you going to do? You make more oil, right? So when you look at this figure right here in the low sugar, this protein here, labeled with W, this is a transcription factor that that actually regulates series of fatty acid biosynthesis genes. Okay, so when you have high level of this W protein, this transcription factor, you make a lot of oil. Okay, but if this protein is not present, if this protein is degraded, okay, so what does it mean? It means that, hey, plant's not going to make any more oil, right? So in this case, we have low sugar. Okay, so these two, this pathway is a little complex, but make a long story short, these are two kinases. Okay, so this protein gets phosphorylated, okay, by this protein, okay, without when you have low sugar condition, okay, this protein, oops, did I just, sorry about that. This protein, okay, phosphorylate this transcription factor. When this transcription factor gets phosphorylated, it gets sent to proteasome degradation. So it's degraded. It can't do its job anymore. In the high sugar condition, okay, this is the sugar. That's the key signal here. This sugar interrupts the interaction between these two kinases. So this guy cannot phosphorylate this protein K here. So when this protein K here is not active, it cannot phosphorylate this transcription factor. That means that this one will not be degraded. This one is stabilized. Then this one can initiate series of fatty acid synthesis.
That sounds like a very, it's a very interesting.
It's very complex, yes.
But in
right.
a very delicate balance. So your low sugar, if you have low sugar, I guess the plant doesn't want to waste resources, right? So your sugar is low.
Right.
So if I don't have enough of that carbon source, I don't need to be using my energy to make something that I don't need. That's the plant.
Yeah.
And so it says no oil. It's a very delicate balance of that event where you have high sugar. It's a lot of nutrients, right? That it's making sugar is abundance, and the plant is saying now we're good to go. We can make oil as much as we need. Is there a way? Is there a time for the plant that too much oil is too much oil? So it is a way that even high sugar amount that
it reaches the capacity. Yeah, absolutely. Yeah. It reaches the capacity. Yeah.
When that happens, so the plant stop, it sends us a bad signal back to just stop that signaling internally.
Right. Yeah.
So you have looking sugars as the carbon source, as the plant is able to do something like this, utilizing other carbon sources aside from the sugar that it naturally makes, as a part of photosynthesis, or sugar is the main trigger of all the molecule fatty acid synthesis.
Well, so fatty acid synthesis is, basically you have two carbon, right? So you start from fatty acid synthase. So yes, carbon skeletons you know, from sugar, from so that's mainly, yes.
That's mainly true. Mm-hmm.
Yeah.
Mm-hmm. I know that you look at plants, and I'm sorry, you look at the seeds and the leaves as and as the way to, you know, increase the production of oil. Production of oil. So you're looking for oil that is just necessarily a oil. You're looking for improve the quality of oil. But before we have that question, there's a question on the chat, Jan, said, do all plants do this the same way? Are there some plants that are unusual in this regard, I guess, in the way the signaling pathway occurs?
You mean making different type of fatty acids, right? Or the regulation?
The regulation. Mm-hmm.
So, well, basically, they are, you know, the same way. So you have certain sets of fatty acid genes involved in lipid biosynthesis.
Mm-hmm.
So they are usually the same sets that would function the same way. Yes, but some plants will make different type of fatty acids. Some plants will make more of certain types of fatty acids, you know, different from other types of plants. Yeah.
Mm-hmm. So so the pathways
and that,
that
of
are
course,
regulated.
that of course will be different sets of enzyme involved
Correct.
in that. Yeah. Mm-hmm.
So the pathways are that regulate this fatty acid or production are fairly conserved then.
Yeah.
They're fairly conserved. That's actually very
Mm-hmm.
good. Do the plants make the same amount or the same type of fatty acid, or can you modify plants to make this fatty acid that you are desiring the most? You know, for a very specific application.
Yeah, so like I mentioned earlier, that, you know, certain types of plants, of course, they some of them make unusual fatty acid, make different other types of fatty acid. The compositions are different. Yes, definitely. So what we're trying to do also is, you know, you alter the composition of fatty acids. What type of fatty acid do you want more in your plants, for example? If you want more monounsaturated, you know, it's a big deal right now that, hey, it's beneficial to, you know, health and everything. So if you want to make more mono or polyunsaturated fatty acid, what type of pathway you're going to do. So basically, you just have to look into the pathway of the biosynthesis, which involve with so many types, so many enzymes, you know. So you just have to see that, hey, okay, if we block this pathway, we knock down this gene, we maybe overexpress this gene, what's going to happen to the pathway of biosynthesis. Yes.
Yeah. And that makes sense that you do use, since these pathways are somewhat conserved, that you use a plant model like a Rhabdopsis, so you understand you have a very well-defined organism. So when you make any changes or any mutations, you can actually know and then take that into perhaps a different, higher plant. You know, with a better
absolutely.
What that means.
Yeah,
absolutely.
Yeah,
that makes sense to do that.
Yeah.
And so one thing that you and I were talking about was that the better the quality, that one of the things that you know, you're looking into, and then maybe talking a little bit about into the future, is the better the quality of this oil that is being made. It addresses some aspects of the climate. It helps into the climate change piece, meaning that the better the oil, we hopefully that as they get processed, the cleaner the biofuel would be. Is that something that?
So basically, we all know, even though we're not driving anywhere right now, so, you know, I mean, so since we home right now, we're not driving anywhere. But the thing is that fossil fuel is going to be gone one of these days, you know, and you cannot renew it right? So why is plant oil so important right now? Because it's renewable, right?
Exactly.
First thing is that it's renewable, it's sustainable, right? Is carbon neutral, right? So when you do photosynthesis, what happened there? So plant takes up carbon dioxide, right? And then it releases the same amount of carbon into the environment. So what do we want? We don't want any more of the greenhouse gases, right? So that we would help climate change crisis that we're facing right now. So we don't want to produce any more of carbon dioxide into the environment. So plants is the answer.
Yes, one of the answers to that.
Yeah.
It's a great application into looking why we do the kind of research we do. There is a question that came up, and so for you, Jan, says, are there any consequences to making plants continuously produce oil?
Consequences.
Meaning that, so the plant is making oil all the time. Are there any good consequences or bad consequences for the plant, for a plant that is continuously making oil? And
yeah, that's a great question. So actually, so the ideal, right? The ideal is that, hey, I want plants to make a lot of oil and also make plant, gigantic, right? So plants are big and also make more oil. So that's always a trade-off. Like, okay, you make more oil, but the plants are small. Right. So that sometimes that happens is a trade-off. Yeah. Yeah.
So they don't grow as expected, right?
Right. Because you are actually messing with their metabolism, right? So when you knock out one gene, it's not going to affect just one thing, one pathway. Right.
It's going to affect many other ones too.
Exactly. When you get rid of one protein, it doesn't mean that, okay, this protein is not going to phosphorylate this transcription factor. No, doesn't mean that at all. It also stopped the phosphorylation of a series of proteins, other proteins that we're not looking for.
And that tells you about how this molecular pathways are interconnected with each other.
Absolutely. That was a great question. Yeah.
There's another great question here. It says, how much more fatty acid or oil is produced when the stop signal is manipulated and removed from the plant?
Can you repeat that again?
Sure. How much more oil is produced when the stop signal is manipulated and removed from the plant?
Okay. So the first slide that I showed there, very interesting. So we got triacylglycerol up to about almost 40% in seeds. Yeah. So that's actually a big jump. Yeah.
That's quite a lot, actually.
Yeah, that is quite a lot. Yeah.
And
the mutant plants actually look normal.
That was my next question.
Compared to the wild type plants. Yeah.
The growth is normal.
Right. But it's interesting that when you try to knock out 3 of these analog proteins, the plants were not viable. So that's the consequence. That's one of the consequences. Yeah.
So I am also thinking that when the plant, going back to the previous question about the plant making constantly oil, also the plant is also using a lot of its resources too, energy resources to that. And even if you're also changing the molecular, the pathway and how they communicate for growth, let's say, the plant is for using that energy, is consumed, for lack of a better word, in that particular pathway, which is making oil, oil.
So but we can't think that, okay, you study plant lipids, you think about plant making oils, that's it, but that's not it, because when you think about lipids, what's lipids? Lipids is the structural components of the cells,
Correct.
right? When cell
Mm-hmm.
grows, you have to make more lipids. Lipids is the energy storage of the cells,
Mm-hmm.
right? And also one thing that people usually forget that is involved in cell signaling
Sure.
system in plants, right? Yeah. Probably heard of jasmonic acid, for example, right?
And
So
it's also
that's one of
part of the structure of the cell too, right? Plasma membrane.
right.
Yeah.
So plants are pretty smart. You think that plants don't talk? They do. They really
Yeah.
do. But they talk in their own language. Yeah.
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 Well, maybe,
Yeah. They send signals. They send signals to each other and said, "Oh my God, there's a pathogen here, so watch out."
Yeah.
They do it their own ways. Yeah.
Correct. Correct.
Yeah.
And I think
Yeah.
and that signal is very interesting and very important. There's one question here in the chat for you. It said, what part of your research findings surprised you the most?
What part of the research?
Of your research findings surprised you the most?
Surprised me the most. Well, I mean, the first slide that I showed you guys there, that that we have the analog protein, the inactive analog protein, is pretty intriguing actually. So, like I said, it's like the invisible break that you didn't know that it existed before.
Mm-hmm.
And then you see this, and we did series of experiments to see that, hey, it's actually incorporated into the complex, even in the normal growth condition. It's always in the complex, just constant breaks. I mean, the plants always put brakes on lipid biosynthesis constantly. If you remove that, then you make more oil.
Like, wow. That sounds, it's true. Sounds like a little bit of when you mentioned, you just said that there was a sense of discovery, right? You just spoke with a lot of excitement about it.
Yeah, I think it's intriguing. Yeah.
So Jan, we're gonna pivot a little bit, and we know we have students and teachers perhaps listening to today's Q&A. We're gonna pivot a little bit to the students that and address them now. So I know that how long have you been at the lab?
5 years now. Yes, 5 years. Yeah,
I remember when we first met you. It's been a while. So you came straight from your graduate school?
Yes.
To your postdoc, right?
Right. Time went by so fast when you're having fun, right? It felt like yesterday. Yeah. So I graduated from University of North Texas in Denton, Texas.
Mm-hmm.
Yeah. Yeah.
And so if sometimes I get this question asked a few times from the students that we see. So did you always know that you wanted to be a scientist? When did you?
I did. Yeah.
You did.
I did. Yeah, I did. When you look around, when you look outside the windows, you see that, hey, how come, in the winter, there's no leaves at all. What happened? Everything turned brown, and then all of a sudden it's green again.
Yeah.
How come, these guys, you don't see them anymore on the ground in the winter, and then all of a sudden they're just something just popping out of the ground, and then they produce flowers. Sometimes, why do leaves turn purple? What happened to them? It's kind of fascinating, and I always wanted to know answers. Yeah.
And so those answers, led you to pursue your graduate career. Would you always be a plant biologist, or there's something the plant biology something that you started to do here? I'm assuming that that's not the case. So you were
No.
Plant biology as well?
Yeah. So I've been studying plant lipids for my PhD degree as well. Yeah.
Very, 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 very cool.
Yeah.
I know that. So for the students that are listening and so forth, if you're a high school student, even as a college student, what kind of preparation would you suggest, of courses that they take, and taking advantage of research internships, for example? What is the value that brings to your career path? Did you do any internships as an undergraduate student?
Yeah, I did. I tried, well, for me, I tried to work with a lot of mentors, try to learn, from a whole bunch of people that had, experience. That's probably one thing that you need to do, definitely. And read a lot, actually, because I know that all the really successful scientists, that I know they talk to, they spend a lot of time each day reading, keeping up with recent articles, new findings. That's one way to keep up with the research world. If you, well, some people think that, well, you are going to be a biologist, you don't need to pay attention to math. That's not true either. Okay. And I do regret that a little bit, that I didn't really pay more attention to math. But you actually use it a lot. Okay. Statistics, you also use that a lot. Yeah. Chemistry, organic chemistry class, that's important too. So yeah.
Right. It's building that in a multidisciplinary platform that then takes you to the next level. It's a very important thing.
Yes.
I know you have mentored students, high school, college, and graduate students as well. What have you learned from being a mentor?
What have I learned from being a mentor?
Yeah. What have they taught you in terms of, sometimes I feel that students do look at things in a very different way.
Absolutely. The students, the interns, that I mentored these past years, which I'm extremely grateful, is a great opportunity for me. I learned that, yeah, absolutely, we look at things from a different point of view, and the most rewarding experience coming out of that is that, well, you might change somebody's perspective. You might change somebody's point of view, that, hey, biochemistry is not that bad. It's actually pretty fun. Yeah. So students, interns, they usually have different expectations. Okay. The most important thing working in the lab, don't give up pretty easily. You're gonna fail. You're gonna fail all the time. Like, seriously, not you're gonna fail all the time. That's why when you get, good results, you say, oh my God, it's fascinating. It's great. It's awesome. But you have to face failures before you can achieve something. Yeah. So don't give up. Yeah.
Learning process.
Yeah.
Jan, I'm gonna just ask you another question. Just came into the chat, and I want to address it so we don't forget. It has to do with the science piece. Is there a simple way to measure oil in plants for a student science experiment?
Simple way.
Could you measure oil in plants for a student experiment? I'm thinking perhaps a high school student looking into doing a plant experiment that looking at oil production or oil inside the plant. What would you suggest the student? What kind of simple ways that they can look into those oils?
Okay, so what we do is, a little more complicated. So you have to actually extract the lipids, and then you derivatize them, you inject them in GCMS and look at the lipid profile and stuff like that. But if you want to just stain them with the lipid stain, and then look at lipid bodies in leaves, for example, under microscope, that would be one way to do. Yeah. So those would be.
Go ahead. Sorry.
Yeah. So that would be simple, but all you're gonna see is that, hey, I see lipid bodies, but you're not gonna know what they are. So right.
But
Yeah,
that's, I think,
but
a
if
very
you,
good way to start counting, at least to see they are, and depending
you're
on what
right.
experiment that they're doing, they're
Hmm.
able to at least get a sense whether this
Something you can do under microscope. Yeah,
correct.
yeah, right, right.
Maybe if you can't, do you have an idea? Maybe if the person contact, maybe we can. Do you have a sense of what that or that oil stain might be? Do you have top of your head? Can you recommend that oil stain that might work?
Yeah, yeah, absolutely. I mean, you can use the Nile red, for example,
Oh,
to stain the
mm-hmm.
more. Yeah, yeah, for staining lipid bodies. Yeah. So that's what we use as well. Yeah.
So we're getting to about 4:41, and we just don't want to, we want to cut up in time. We all through our scientific careers in life, we have had mentors that have guided us, right, through this. What would be the best advice that you received from a mentor? What was the best advice that you received from a mentor?
I'm sorry.
What has been the best advice that you received from a mentor?
The best advice that
That a mentor
I've
gave you?
gotten
Mm-hmm.
from my mentor,
Yes.
the best advice that I got from my mentors is that, just work hard, be persistent. If you fail, you fail, and then just,
Learn from it,
try something
right?
different, look at the different pathway,
Mm-hmm.
yeah, yeah. So you just, working in this field, you just have to be extremely persistent. You never ever don't give up. It's going to take a long time. It's definitely going to take a long, long time to achieve something. Definitely.
Yeah,
Yeah.
I would say it's more of a uh, it's a marathon. It's not a sprint race, right?
Yeah. It's not a sprint. Yes, absolutely. Yeah. Yeah.
So there's a question. I'm not sure we may have answered or not, but I just want to make sure I will share this one with you. What advice would you give to students who are in college and that will probably graduate and will start going into a working career in research? What advice did you give a student into perhaps looking into research as a career?
What's the advice that I would
If
give
you-
to?
Mhm. A student that might be thinking into a career in research.
Um, Make, if you get a chance to go to meetings and stuff like that, make connections. Okay. If you want to work in particular labs, for example, scientists that you're interested in their works, okay, try to read about their works. For example, study what they're doing in the lab, and then make connections. Try to talk to them and see if there is any opportunity somewhere.
Mhm.
That would be my recommendation. Yeah.
That's
Yeah.
a very good advice. And I think for those that are thinking even to go to graduate school and after graduate school, something that, I did my PhD back in the 90s, so it's a different time. And now that you have to do postdocs, so part of your thinking, your career plan is including that type of that's training as well, the postdoctoral
Mm-hmm.
time. Okay.
Yeah. Yeah.
I don't think we have any more questions. I'm just looking here. So, Jan, thank you so very much for
Thank you
joining
so
us.
much for having
It
me.
was a
Yeah.
wonderful. I told you this before. I'm a fan of what you do. I think
Oh,
biologists
thank
are,
you.
and I'm hoping that, we convince a few folks out there that this is a field to get into, that is a field to be curious about and a field to explore.
Yeah.
I think it would be very good. So thank you for your time. We
Absolutely.
appreciate the work that you do. Then our next, and let me see if I can actually manage the the uh, the sharing of the screen sharing thing here. Let me do this very quickly here. So our next science share will be on On next Science Thursday, I hope everybody can see this. Our next, which is
Can
probably
see
not,
it.
you can see it. Our next Science Thursday it's on July 16, and we will be honored by having Vanessa Sanders and Jasmine Hatch for the radioisotope group to share with us about radiochemistry and the applications of radioisotopes and radiochemistry in medicine and imaging. And if you have any questions about it or any concerns, please, any questions, please let us know. We should be posting our event for the announcement for the next July 16, 2020 Science Thursday on the Office of Educational Programs webpage. Again, thank you so very much, and I would like to thank Brookhaven National Lab for hosting the event today, the Office of Educational Programs, my colleague Diana Murphy, and I encourage you to check the programs that we do at the OEP and at BNL. Thank you so very much.